first question is going to be to Victor. What is Down syndrome and what is it caused by?
Down syndrome is a condition that's caused by an abnormal number of chromosomes. So our genes in every cell in the body are clustered on structures known as chromosomes. And in humans, we have 46 chromosomes. There are actually 23 pairs of chromosomes. So you have 2 copies of chromosome 1, 2 of number 2, 2 of number 3, etc. And in Down syndrome, instead of having 2 copies of chromosome number 21, we have 3 copies. So there's an extra chromosome. So instead of 46 chromosomes, there are 47. And having that extra chromosome is clearly what causes the syndrome. The syndrome itself is very complex. So people with Down syndrome can have a whole range. It can have a whole range of effects. Many of you will be familiar that learning difficulties are a major part of it. We're going to be talking about the early onset Alzheimer's disease. Already mentioned congenital heart defects. Obviously, very characteristic changes in the shape of the face and the hands. Increased rates of leukemia. So the whole range of conditions, and actually conversely, increased rates of leukemia, but people with Down syndrome also have lower rates of solid tumours. So there are some positives, if you like, to it as well. So that's what causes it, but that doesn't really get at the heart of what's going on. Why is it that an extra chromosome would cause all of this very diverse range of effects? And so what we do know at the moment is that this chromosome contains a number of genes. Depending on how you count a gene, the number is around 300. So there's around 300 genes. It's about 1% of all the genes that are in the human genome. So it's not very many of them, but it's 300. And we think it's those 300 genes, when present in one extra copy, instead of 2 copies, we have 3 copies of it. That's what's causing these different phenotypes, these different effects. But what we don't know at the moment is which of those genes is responsible for which of those phenotypes. And I think we'll get to discussing that in a later stage.
Thank you very much. So that's what causes it. Andre, I'd like to come to you to talk a bit about what life is like for people who have Down syndrome.
Well, many people with Down syndrome actually have a very good quality of life, enjoying going out with their families, enjoying various activities. I work in a clinic for people with learning disabilities, and so we see some people with Down syndrome who, for example, work in shops. Other people who are taking part in our research are very talented and are artists or actors, musicians as well. And so there's people can have a very, very good life. But as Victor has already mentioned, some people have more difficulty than others. And we now know with the increase in life expectancy that some people age faster, or most people with Down syndrome would age faster than people in the general population. And in our clinic for people who have memory problems, we sadly see some people developing Alzheimer's disease at quite a young age, in their 40s. But we also see some people who reach the age of 60 and who do not have any significant difficulties. So there's a lot of variability, and we're quite interested in understanding why we see that kind of variability.
And could we say a little bit more about what you mean by age faster? So you're getting to the point where you're having seeing symptoms of things like Alzheimer's disease earlier, but there's...
Yeah, so some people think that people with Down syndrome develop some, not just Alzheimer's disease, but some of the other aging-related conditions a bit faster than in the general population. So you see grey hair, like myself, at quite a young age.
Balding? Was that?
Not necessarily. Skin aging, perhaps.
Okay.
So there are some signs of faster aging in general, but as Victor mentioned, people with Down syndrome are protected against some conditions, such as solid tumours, some of the cancers, and we also think that
what is that?
Solid tumours are the sort of common cancers that people develop with aging, like say lung tumours or colon cancer, those kind of things. And the other thing that's good is that people are also relatively protected against ischemic heart disease. So,
and what is ischemic heart disease
so
versus?
so that's a kind of heart attack which is caused by a clot in the heart, and may also be less likely to have high blood pressure.
Okay, so it's an incredibly complex sort of range of conditions that are linked. And you're all part of the Lundowns Consortium, and that's specifically looking at this one aspect of the link between Alzheimer's disease and Down syndrome. So I'd like to come to Lizzie. Could you talk about some of the areas of research that you're doing?
So Victor and I have had a long-standing interest for unbelievably about 26 years now for working on Down syndrome. And we realised from our point of view as people who work with mice that model aspects of Down syndrome, and I expect we'll get into this a bit more, that there was some very interesting biology to do with understanding the link between Down syndrome and Alzheimer's disease. And we have come together, funded by the very wonderful Wellcome Trust, with 4 other groups. It's because science doesn't work in a vacuum, and the best science is done collaboratively, where you can share ideas between people from different fields and bounce off one another and understand what's going on in different fields, so that the sum is greater than the parts, if you like. So we're part of the Lundowns Consortium. We have a website. It's completely devoted to understanding the link between Down syndrome and Alzheimer's disease, but we're made up of different components. So Victor and I provide the mouse component. We're working with mouse models. We're making mouse models to try to understand the biological link between Down syndrome and Alzheimer's disease. We also work with Andre, who is in fact our leader, who is a geriatric psychiatrist. So he's not a geriatric psychiatrist, you're quite young actually, but a psychiatrist trying to study or studying people with learning difficulties in older age. And so he's looking at when people with Down syndrome develop Alzheimer's disease and characteristics about those people. We also work with someone called Annette Karmeloff-Smith, who's a very talented and very, very distinguished expert in childhood development. So she's trying to look at the links of what might there be in early childhood that's different about people who develop the early onset Alzheimer's disease in Down syndrome compared to the relatively few people with Downs who are protected from Alzheimer's disease. And then finally, we work with 2 other groups. One is a group headed by somebody called Dan Nizitek, who's working at Queen Mary University in London and in Singapore, actually, who's taking cell lines from people with Down syndrome and trying to look at cellular changes, again, to understand if you have Downs, why is it that you will then have a greater propensity for Alzheimer's disease?
So you mentioned protected groups just a minute ago. Does that mean there are some people who are less likely to get?
Yeah, so if I just introduce the last group, because
sure.
he's very famous. So you might just have seen, especially if you've been watching Newsnight, that there's a person called John Hardy from London who's a London scientist. He's just won an absolutely whopping, enormous prize funded by Mark Zuckerberg from Facebook and various other people from Palo Alto, because he developed one of the first theories about how Alzheimer's disease arose. And so he's now working at UCL, he's back from the States, he's working at the Institute of Neurology, and he's a human geneticist. So he's combining his expertise in human genetics, our expertise in mouse genetics, Andre looking at older people with and without Alzheimer's disease who have Down syndrome, Annette who's looking in early childhood, Dan who's looking at cell lines. We're all coming together to try to understand what is the biological link between Down syndrome and Alzheimer's disease. And that's really important for 2 reasons. Firstly, because the single biggest risk factor in the population of getting Alzheimer's disease is having Down syndrome. And secondly, for the rest of us who don't have Down syndrome, what we learn about Alzheimer's disease from the Downs population is informative for the rest of us. It tells us about biological pathways that we really need to know about to understand neurodegeneration. And I've completely forgotten what you just asked me.
So it's just that you made a point about some people are protected from Alzheimer's disease, and so I was wondering whether what that means. Is that the people who don't go on to get Alzheimer's, or is that also telling us something about?
No, that's a really good point. So what we know is that if you are able to look in the brains of people with Down syndrome, pretty well universally, by the time those people are in their 40s, they have the chemical changes. We can actually see the chemical changes in the brain that are absolutely diagnostic, if you like, for having Alzheimer's disease, but they don't necessarily have the dementia. And this is a strange and interesting thing. And you can draw graphs and you can look at, say, people who, we have people we know about with Down syndrome who are in their 70s and 80s. Presumably their brains are full of the kind of changes that one sees classically in Alzheimer's disease, but those people are absolutely well. They do not have dementia. So what that's telling us, and we have some preliminary data from our mice, that in this very complex picture that is Down syndrome, there are likely to be genes on chromosome 21 that, if you've got a particular combination of them, may even protect you from having Alzheimer's disease. And again, that's a very important finding for understanding what Alzheimer's disease is and ultimately how to treat it in all of us.
That's amazing. So it's actually the same syndrome. It's in some people is causing an increased risk, and in other people causing a decreased risk.
Yeah, and that's one of the take-home messages about Down syndrome. It's incredibly variable, so difficult to investigate.
So I'd like to ask sort of how working with mouse models came about. Why study Down syndrome in mice, and also what is a mouse model of Down syndrome?
It started with a chat in a pub, actually, somewhere in Bayswater. We've been debating which pub it was. We can't remember, because it was quite a few years ago. We basically came up with some ideas about how we might use mouse genetics to try and address what's going on in Down syndrome. And I think before I just talk more about the mice, the question is, why can't you do the sort of genetics that I'm about to discuss? Why can't we do that with the humans, with humans who have Down syndrome? And the answer is you can, but only to a limited extent. So one of the things that we're very interested in is understanding, I think I mentioned this earlier, 300 genes on chromosome 21, all present in an extra copy. Which of those 300 genes do you need to have an extra copy of in order to have this increased risk of Alzheimer's disease, or the congenital heart defect, or the increased rate of leukemia, or the learning difficulties, any of these things? We're trying to identify which of the genes this is. Now, could you do this using human genetics? Well, you might be able to. So there are occasional rare individuals with Down syndrome who don't have an extra copy of the whole chromosome. They have an extra copy of just part of the chromosome. We call them partial trisomies instead of the full trisomy. And then if you look at which bit of chromosome 21 they have an extra copy of, and ask which of the different aspects of the syndrome do they have? Do they have the heart defect? Do they have the leukemia? Do they have the learning difficulties? You could map which region of the chromosome is responsible. But the problem is that these people are rare. There's not very many of them. And you're essentially limited to what human genetics throws up at you. So the variation that's there, occasionally an individual does have this partial trisomy, but exactly where that partial bit of the chromosome is, you have no control over.
Is there variation as well within, if you get different parts?
Well, you don't get the same. You don't ever get people with the same partial trisomy. So you can't compare, 100 individuals with exactly the same partial trisomy and ask, do they all get learning difficulties, or do they all get congenital heart defect, or none of them? You can't do that. You just get solitary, individual, anecdotal, if you like, individuals. So that's what people have done with the human genetics. Lizzie and I thought, well, we could try and address this using mouse genetics. So why the mouse? Well, the mouse is evidently a mammal. It's related to humans. 80 million years separate us from the mouse in evolutionary terms. It's a long time, but not that far in evolution. We share, 95% or more of the genes that we have. The mice have as well. Obviously, they're slightly different genes, and they work in slightly different ways. But there are so many parallels in the way mouse physiology is set up and human physiology is set up that it's a worthwhile model. It's also, of the mammals, it's by far the best developed genetic organism, meaning that we can alter the genes in the mouse essentially at will. It can take a long time, it's very slow, but essentially we can make almost any modification that we can think of, we could eventually make. And that makes it a very powerful tool. And so this chat in the pub basically started with, well, could we use the mouse to model Down syndrome? Okay. well, how might we do that? Well, is one of the mouse chromosomes equivalent to human chromosome 21? If it is, we'll just make an extra copy of that. Well, there isn't. The genes that are on human chromosome 21 just happen, through evolution, to have been shuffled into at least 3 different regions on 3 different mouse chromosomes. So that makes it a bit awkward. So we thought, well, maybe what we could do is to move the human chromosome into the mouse. We make a mouse with all its normal chromosomes, and then 1 extra on the human one. So that was an idea over a beer. 13 years later, we published the paper. So it was slow for various reasons, but we did make a mouse that did have this extra chromosome. And
And that was a human.
this was the extra human chromosome, yeah. And it turned out this mouse had learning difficulties, congenital heart defects, certain signs of kind of early aspects of leukemia, etc. So that was sort of the first stepping stone. And then since then, we've developed a whole set of different mouse models, all containing different sets of genes in 3 copies. And we're using those essentially to map and find the individual genes that cause the different phenotypes. And in my own group, we're focusing on heart defects. You're working more on the neurodegeneration, for example.
And so why have you picked 3 different versions of the 3 different sets of genes? Did I get that right? There's 3 different mouse
Well, the mouse
chromosomes.
equivalent genes, the mouse genes, the equivalent ones to the ones on human chromosome 21, are dispersed, if you like, in 3 different blocks. So another way to do the experiment is to make duplications of just those 3 regions, but you have to do that 3 times and then breed it all together. This has been done as well, and we're part of the groups that are doing that.
Is it still the sort of 300 genes that we're talking
Yes.
about here? So
Well, so initially we've made mice that have all of the genes extra, but now, as I said, we've made a whole, a colleague of ours calls it the zoo. We've made this little zoo of different mouse strains that contain, some of them as few as 15 genes extra. So if one of these strains that only has those 15 genes in 3 copies instead of 2, everything else is completely normal, and that strain were to have, for the sake of argument, learning difficulties, we would know that somewhere within just now those 15 genes, there must be at least 1 gene that is causing the learning difficulty. So that's our strategy. So for the congenital heart defects, for example, we've managed to map our way down to a minimum region of 39 genes, and within that we know that there's more than 1 gene. When we break it up further, the heart defects disappear. So we now know that there's at least 2 genes in that area, and we're hunting for them, but haven't found them yet.
So you're trying to make a model where you have the different types of characteristics that appear in humans with a limited number of genes, and trying to find the cross-section between the genes which give you the symptoms or the conditions.
So conceptually, we start with adding an extra copy of all of the genes,
Yep.
and then we get mice that have all sorts of different phenotypes, the learning difficulties, the heart defects, or whatever. And then we can make ones that only have an extra copy of smaller numbers of those genes. So instead of 300, only 100, or only 50, or we can break it up into chunks, 6 chunks of 50 genes each. Conceptually, that's sort of what we're doing. And if we say, okay, the second chunk in from the left is the one that causes the heart defect, we know it's somewhere in there. And then we can break that up into 5 chunks each of 10 genes.
And so
and
you're
do the
looking for the
experiment
chunk
again.
where you get the symptoms of Alzheimer's.
Yeah, because unlike, probably many of you have heard of various genetic diseases like cystic fibrosis or beta thalassemia, or many of these conditions. These are caused by genetic changes in genes, mutations in genes, and today's kind of DNA sequencing technology means that actually you can quite quickly sequence the whole genome and find those mutations. Down syndrome isn't like that at all. It's an extra copy of a completely normal gene. It's not a mutant gene, it's just an extra copy. Instead of having 2 copies, which we normally have in most genes, we have 2 copies, we now have 3 copies. So just that increased dosage is what's driving this. And so we can't sequence our way to the mutation. We have to use some other approach. And so for us, we're using the power of mouse genetics, where we can alter copy numbers of genes at will, essentially, to map our way to finding the causative genes.
Yeah. So I think I've got that. So yes, we've got a question here.
Yeah.
The leukemia that somebody with Down syndrome may get, has that not got it? I just want to clarify, it doesn't have the same genetic cause as leukemia in somebody without Down syndrome. Is that kind of what you're saying?
Yeah, they get a rather rare form of leukemia. It's called acute megakardioblastic leukemia, and that's very rare in the non-Downs population. And in fact, if you look at the genetic changes in that particular leukemia in people who don't have Downs versus people who have Downs, the other genetic changes are different. So although you end up with the same sort of disease, it's actually genetically it's being driven by it in a different way. So they get a rather unique form of leukemia. Yeah.
And does it apply to the other conditions like ischemic heart disease that Down syndrome people are prone to? But is that different to...
They're less prone to ischemic heart disease, I think.
All right,
It's the congenital heart defects. This is babies are born with defects in the way the heart is formed. There are holes in the heart, essentially, between the different chambers.
Yeah.
So it's a problem with the way the heart develops in utero, and it doesn't properly divide up into the four chambers.
Yeah.
Our hope is that by understanding what is going wrong in the Downs case, we will get an insight into pathways that might be perturbed in the non-Downs population as well. Of course, it doesn't have to be that. I mean, you could end up, there could be two different ways to get to the same point.
Yeah.
You could get the same condition, a hole in the heart caused by two completely different causes. In the Downs case, it's caused by, pathway 1, and in the non-Downs, it's caused by pathway 2. But it's certainly a route in, and it's certainly something we're very aware of, and particularly when we get to talking about the Alzheimer's disease, I think, there we're talking about obviously a major health problem for the non-Downs population as well. And insights that we get from the Downs population, we would hope to transfer to the non-Downs as well.
Hi, I was interested to hear about the learning difficulties and how that translated across. I mean, obviously there are many different types of learning difficulties, I don't know, cognitive, linguistic, or what have you, but how do you map that across? Yeah, how do you map that across to a mouse?
Well, mice learn. So obviously mouse behavior and human behavior is different in very obvious ways to us. Mice don't read, they don't, that sort of thing. But they certainly learn. They have spatial memory. They're different, they're nocturnal, which we tend not to be. But we can test aspects of how they learn quite readily in terms of spatial memory in particular. Olfaction, they're very good at sense of smell, so we can measure their ability to remember particular smells. We also work on other aspects, so locomotor function, that's the ability to kind of, move your limbs in the correct order at the correct time. And there's evidence that people with Downs have impaired ability in locomotor function. Some of our mouse models also have exactly the same problem. So, we can test that quite easily in the lab. We can stand them on a little kind of broom handle, if you like. Put them at the end of the handle. They don't like being out there. They don't like being exposed. They worry about predators. So they have to scamper back along the thing, and that's, a little bit of a balancing act. So we can time how long it takes them to get back, and that's one way to measure it. Or we can measure put them on a wheel and measure how fast they can go around the wheel. And we can make the task harder. We can take a wheel and take some of the rungs out.
Lots of different tests. So actually, I'd like to come to Lizzie next, and this may be connected as well to how you find out, or how you test. So you've got the learning test, but then how do you test for symptoms of a mouse having symptoms of Alzheimer's disease? And what do we know about, sort of from all these models, what genes are involved in the Alzheimer's disease?
Just to come back to your question, it's actually a completely critical question. It's a key question. It's a question that we're funded to address, that's becoming increasingly important in understanding what it is to be human. And that is the issue of how the interaction between environment and genes and inheritance, all the other things, what you get to think of that affect our behaviour. So obviously, when we work with mouse models to understand Alzheimer's disease, we want to look at a mouse and say, well, if we think you're coming down with a mouse form of Alzheimer's disease, what happens to learning and memory? And so, as Victor said, there are very sophisticated tests of learning and memory in a mouse, and they're getting increasingly sophisticated. Those have been there for many years, But then the next question is, how do you relate that to what's going on in the human condition? How do we draw parallels between the two? And the short answer to that is it's bloody difficult. But fortunately, we work with people like Andre and Annette Karmeloff-Smith, who are experts in human behaviour. We also work outside with other groups, particularly groups in Cardiff and Oxford, who are very good on mouse behaviour. And so we have regular meetings between the mouse behaviouralist, the human behaviouralist, to try to align, if you like, human psychology with mouse psychology, mouse behaviour. And it's very difficult, and from an outsider's point of view, and I am an outsider, actually really interesting to see the commonalities between our two species, and of course there are species differences as well. So it's a fundamentally important question, and thank you for asking it. It's a good one to ask. So coming back to the genes, the person I mentioned who's just won the big breakthrough prize, John Hardy, who's won it for his original insight into the causes of Alzheimer's disease, and with whom we work, who's part of our consortium. Some years ago, many years ago, in the early 90s, he said, and this was data from human cases, human families with early onset Alzheimer's disease, that means full-blown Alzheimer's disease with dementia before the age of 60, that's what we're talking about. So in those families, he found that there is a gene called APP, that's just the name of the gene, and it gives a particular set of instructions to make a protein in the brain, that's all it does. But when that gene has a mutation in it, when it has a change in the DNA signal, it gives out a slightly different instruction. And for reasons that are still not absolutely clear, that is what gives you early onset Alzheimer's disease. So that's one piece of interesting data. That's why John has just won megabucks from Mark Zuckerberg and other people for this insight, and it's a very, very important insight. Then it turns out that that particular gene, just that piece of DNA that gives out an instruction to make a protein, that's all it is, that little gene sits on human chromosome 21. So the idea has always been, and there is other data to support this, that the reason that people with Down syndrome get early onset Alzheimer's disease is because they have 3 copies of that gene, not the normal 2, and we know that having 3 copies is bad for you. That gives you early onset Alzheimer's disease. So, hey presto, we think we've solved the issue of why is it that people with Down syndrome have an increased propensity for Alzheimer's disease. Oh, it's because they've got 3 copies of that gene, APP, churning out more protein perhaps than they need. But it turns out, actually, it's not quite that simple. And we know, we know, as we've just mentioned, that not everyone who has Down syndrome gets dementia. So that's the first piece of information. We know from looking at humans with Down syndrome, not everyone is going to get dementia, even though they've got 3 copies of the APP gene. So maybe there's something protective going on. And then from some experiments that really it would have been impossible to do without the wonderful world of mouse genetics, the sort of thing that Victor and I do, we've also found out that there are genes along the rest of chromosome 21 that modulate the appearance of Alzheimer's disease. So we don't know what those genes are, but what we can say is everything with Downs is complex. It has complex answers because we're dealing with many, many genes that are extra in individuals. APP is a key gene. APP absolutely is what gives an increased risk, but that risk is modulated by other genes up and down chromosome 21. So that's really combining work with human genetics that John Hardy has done with the work that we've done with mouse genetics.
And you mentioned that sometimes there's mutations in this gene. So is there a difference between the different people with this gene, like in Down syndrome cases? Or are you
in Down syndrome, everyone has a completely normal copy of the gene. It's not mutated. Okay. It's just that they've got 3 copies of it because they've got 3 copies of chromosome 21. But we know that having an extra dose of the gene, if you like, is enough to give you early onset Alzheimer's disease. And I think as far as people can make out, and this is a generalisation and therefore not strictly true, if you compare people with Downs to other people who don't have Down syndrome but who have a mutation, who have a change in the DNA message in that gene, actually they're pretty similar. They also get early onset Alzheimer's disease. There are some details there that I'm not quite going into, but that's more or less the case.
So sticking with variability in people, let's come to Andre. Presumably you see this variability with the people you see in the clinic. How do you measure and compare people with Down syndrome and diagnose Alzheimer's disease?
Well, first of all, I should say that just to remind people that Alzheimer's disease is essentially a sort of initially a memory problem. So people develop a memory problem, which then progressively gets worse, and as the disease progresses through the brain, it affects other brain functions and other abilities, and you see a sort of a slow decline. People with Down syndrome present very similarly when they present with Alzheimer's disease, but there are a few differences. So we also see the memory problems, which are common in this population. So when we start to see memory problems develop more than memory problems that they may have had before the onset of Alzheimer's disease, then we begin to worry a little bit. The other thing that's a bit different, though, in Down syndrome, in people with Down syndrome, is that they seem to develop a personality change and quite early on as they start to develop Alzheimer's disease. So become a bit more tearful, perhaps, or irritable, or mood changes very quickly. And some people may also develop epilepsy as the illness progresses, which we also see in Alzheimer's disease in the general population, but it seems to be more common in people with Down syndrome when they develop Alzheimer's disease. So we know what the picture looks like, and that makes it easier for us to pick it up when people start to show these changes. What's difficult for us is because most people with Down syndrome have a degree of learning disability, and therefore have some memory problems already, perhaps. It means that it's difficult to know when you do see changes, whether that's a change for that person. So we always have to establish whether it's definitely a change for that person first before we can be sure that it's a new thing that's developing. And of course there may be lots of other changes, other reasons for these kind of changes as well. So people may have some sort of physical health problem, or they may be having low mood or depression, or something that's upset them in their day-to-day life. So we need to try and exclude all of those things first before we can say that somebody has Alzheimer's disease. From the research point of view, we are very interested in what we call the typical cognitive profile of Down syndrome. So we know young people with Down syndrome have some memory difficulties, but they also seem to have difficulty with some of the frontal functions. So the kind of the executive functions, it's also called, the part of the brain that controls our thinking to some extent. So they seem to have some difficulty with that. And so in our research, we're very interested in measuring those kind of functions and in seeing how that relates to development of Alzheimer's disease in people with Down syndrome. Because we know if people have frontal difficulties, then they do undergo, or if they have frontal deficits, they do start to undergo the kind of character changes that we see in early Alzheimer's disease and Down syndrome. So we can show you some of the things that we do. The type of things that we do is not exactly the kind of test we do. It's a very simple thing. So this is called, I don't know if some of you may be psychologists or have done psychology at school or university. This is called a Stroop test. So it's a very well-known psychology type of test, and it measures a type of executive functioning, so our sort of executive control, if you want. It helps us it measures the ability to remove your first response or remove the noise as well, and make you focus on what you need to focus on. So we'll try it. This is a test that's designed to measure that kind of ability. So we can try it. First of all, in this middle line, I want you to just read what it says. So just read the name of the color. So yellow, black, blue, gray. Okay, so that's quite easy. Next, I want you to say the name. The color is the word is written in. So not what it says, but what it's written in. So let's start with this one. This one. So yellow, green, and red. Okay, and let's try
Black, green, yellow, black, blue, grey.
Ah, you're very good at it. But did you feel that? Did you feel that you had to stop and think? You had to stop yourself for a second, and then just think before you had to suppress your first response in order to say what we instructed you to do. So that's the kind of thing we measure. And some people with Down syndrome find this quite difficult. But you can also tell that we wouldn't be able to use this test with people with Down syndrome because many of them don't read. So we have to think of new ways of doing this kind of tests. So part of my work is to develop the kind of tests to do this kind of thing.
So we're going to sort of explore a bit more with what Andre was talking about before. So could you talk a little bit more about what could be responsible for some of Down's individuals developing early onset Alzheimer's and others not? So just in a bit more depth.
Well, actually, I think Lizzie has already mentioned some of the issues. I mean, we think in our research program, we very much focused on the genetic factors. So we are very interested in knowing, for example, why it is when almost everybody with Down syndrome will have triplication of the APP gene, but not everybody seems to develop Alzheimer's disease at the same rate, and some people are protected as well. So we think that there may be other genetic factors on chromosome 21, but also in the rest of the genome that could help to protect some people. But there may be other things as well. There may be environmental factors too that would be important to explore. So we know in the general population, for example, that you can increase your brain fitness by keeping active, keeping busy, keeping your mind going. And if you do that, then you can help to reduce the impact of Alzheimer's disease to some extent. And the same sort of things could be relevant to the Down syndrome population.
Is that part of what is your wider group looking at these lifestyle factors
yeah,
as well?
To some extent, we are measuring some important lifestyle factors. Yeah, of course, it's very difficult by the time people are in their 50s to know how many years they've spent in school and how active they've been throughout their lives. So it's quite difficult to measure, but we are trying to do make some of those measurements. And the other things that may be relevant is in the general population we know that there are vascular risk factors as well that impact on the risk of Alzheimer's disease. So people who smoke may be at higher risk, people with diabetes because it affects their brain vasculature to some extent. And so on. People with heart diseases, ischemic heart disease, are at higher risk. And people who get stroke, for example, are also at higher risk for developing Alzheimer's disease. It will be interesting to know to what extent those kind of factors are relevant to people with Down syndrome as well.
And when you've got such a complicated set of factors that may or may not be partially responsible, Is the number of people that you study, is that a factor in terms of trying to make these links?
Oh gosh, yes, that's a very important question. We run a very big study. So we have recruited more than 400 people with Down syndrome taking part in our study, but that's not nearly enough people. And so at the moment, the issue is going to be for the future. One of the issues for the future is going to be how to link with other research groups across the world, really, so that we can get enough information on an international scale, not just in a London scale, is also very, very important.
Okay, so I will come back to this collaboration, because it's clearly, even within your group, collaboration is massively important and outside it. I'm going to come to Lizzie there. So you've talked about the lifestyle factor, but within the genetics, so we've got this sort of hammering home this and just making sure I've got it crystal clear. So we've got the 3 copies of the gene, the APP gene, that in some cases causes Alzheimer's disease, some cases, but it doesn't always. And you've talked about other things that might be impacting on that. So how are you investigating those other genetic factors and how they're affecting that APP gene?
We know that the whatever happens in Downs modulates the effect of the APP gene. It's due to other genes, but we don't know which other genes, and we can tackle the problem of finding out which other genes is it through two different approaches. One is through working with the different mouse models that have different parts, if you like, of human chromosome 21 or the mouse equivalent, and saying, well, if we put that little bit in there in an extra copy, how does that affect the biology of Alzheimer's disease? If we put that little bit of DNA there in an extra copy, how does that affect the biology of Alzheimer's disease? And so we do what we call mapping. We map to a region which has an effect, and we ignore the regions that don't have an effect. So that's through the wonderful power of mouse genetics. And I'm not going to suggest for 1 minute that mice are mini humans. They're not. But what they give us is an animal that we can modulate the genes in that animal very, very precisely, and that allows us to look at the biology. So it's not a perfect system. We're not saying this is human biology, it's mouse biology, but because mice, as Victor alluded to at the beginning, are our distant cousins, separated by a mere 80 million years of evolution, which is a bagatelle in evolutionary time, that enables us to learn about human biology. So mice teach us about the human condition. We can also work with biochemistry. So we can, and in fact we are doing this in our studies at the moment, we can say, well, we've got the condition of Down syndrome, we've got the condition of Alzheimer's. If we look at the biochemistry, the chemistry of the cells, the cellular pathways, what's different between those two? What is it about having Down syndrome, having those extra genes, that changes within a cell? And if we can find something that changes the composition of the cell, the shape of the cell, where the bits and pieces that within the cell lie, then that may give us a handle on what's going on in Down syndrome versus Alzheimer's. So there are many, many different approaches that we can take, genetic, biochemical, to really understanding the difference between having Down syndrome and having Down syndrome with Alzheimer's and having Alzheimer's. And it's the differences that are informative.
And so that looking at it within the cell and the biochemistry, that's still in the mouse model?
Well, we do this in mouse models, so we try to understand the biochemistry within our mouse models, which have very, very precise genetic changes made by us. Our collaborator within our Wellcome Trust-funded consortium, the Lundowns Consortium, Dan Nizitek, who works at Queen Mary's, works with cells from humans. That sounds a bit, oh my God, but actually all he's doing is taking perhaps a very small skin sample or even just plucking out a hair, and we can grow cells from those tiny, tiny, tiny biopsies. Imagine just plucking out a few hairs, growing cells from the bits of skin that are at the bottom of the hairs, and then he can grow large numbers of cells, and we can look at those cells again to try to answer this question: what is it about Down syndrome, about Downs with Alzheimer's, about Alzheimer's? What are the similarities? What are the differences that are informative of the biochemical pathways?
Is the biochemical pathways in the cells growing from your skin the same as what's happening in the brain of somebody? And so just, because you're looking at Alzheimer's.
I wouldn't say it's exactly the same, but it's like the mice. It's a model system. So the cells that we grow from people, from any one of us, we could just give a few patches, a few hairs, a bit of skin to grow these cells. They would model much of the biology of us. But of course, ultimately, the reason that we work with mice is because it's not only about individual cells or tissues, it's also about the interactions between different organs. So it's not, in Alzheimer's disease, it's likely to be not just all about the nerve cells in the brain. There's also other factors such as blood flow to the brain. There are factors to do with inflammation that we think are involved in Alzheimer's disease. So looking just at nerve cells.
Lifestyle factors.
Absolutely. So as Andre said, if you have type 2 diabetes, for example, you have an increased risk of Alzheimer's disease. And it's not, I think, entirely clear why that is, but that's why we, Victor and I, need to work with the whole animal, because we need to bring the studies from the biochemistry, the cells, back to the whole animal, and then ultimately going back to humans through Andre and his studies.
So I'm going to come back to Andre actually to talk specifically about cross-collaboration, because as a sort of key part of CRIC and what CRIC hopes to do going forward. So could you talk a bit more about how your teams have collaborated together?
As Lizzie has already explained, we different teams, but we work very closely together. For example, I lead on the adult team, so we work with adults with Down syndrome, and we inviting lots of people with Down syndrome, adults between the ages of 16 and all the way up to the 70s, to take part in the project. And then we do lots of assessments, cognitive assessments, to understand their sort of cognitive profiles, and also collect blood samples, if people are willing to do blood samples, for genetic analysis. But if they don't want to give blood samples, we can take sputum samples. We take hair samples and give it to Dan for doing some of the cellular work.
So is that connecting up? So that's connecting.
So that's connecting with the genetics stream and connecting with the cellular stream. But at the same time, we're working closely with a group at Birkbeck University who are working with infants and babies with Down syndrome. And so what we had to do was to look very carefully in at the kind of assessments that we do to make sure that they're very similar in the babies and in the adults, which is a very difficult thing to do. So you can't do the same kind of memory test in a baby that you have to do in an adult. So it's quite difficult to do, but we've tried to map it very closely together. And also working then with the mouse model stream, so when they do behavioral experiments, we're pretty sure that they're looking at the same things that we are looking at.
So it's checking that the models, the mouse models and the genetics are mapping appropriately across, so that's where you kind of need to work together. Other sort of things that have come out of this, other collaborations, Lizzie, I think there's...
So one of the other things that's come out of this is... Again, as we sort of have tried to emphasise, what we do is completely collaborative, but not only within our group, but also outside. So we collaborate worldwide. And one of the things that's come out of this collaboration is that people with Alzheimer's have seizures. People with Down syndrome are at an increased risk of epilepsy. So we're not epilepsy experts. Well, actually Andre is to some extent, but Victor and I certainly aren't. But we have this incredibly powerful system where we can start to analyse at a behavioural, at a brain level, at a cell level, at a molecular level, the biochemistry within pathways, what's going on with Alzheimer's disease. But of course, we can also apply that to other conditions, and the condition that's really relevant is epilepsy. And as it happens, there is an extremely good group at UCL, where I work and where Andre works, who are world experts in epilepsy. They are headed by a couple of clinicians who see people with rare and exotic and very, very difficult to treat forms of epilepsy. But they're also very good at working with mouse models. And so these clinicians are extremely important people because they know their way around human clinical cases of epilepsy, but they also know what to look at in a mouse. And so those are ideal people for us to partner with, and we are partnering with them. Victor and I, again, have raised some money from the Wellcome Trust, and we've appointed, from our funding, somebody to work in the lab of these clinicians who work on human and mouse epilepsy, and what they're going to do is to try to follow up the causes of epilepsy in Down syndrome and how that relates to Alzheimer's disease, working with the various models that we provide, but also with their expertise. So that's a fantastically interesting spin-out that's come out of all of this.
So to come to Victor, so talk a bit more about sort of other areas of research that you'd like to see sort of coming up next, sort of what you sort of hope for the future.
Hope in the future, yeah.
Yeah, so the next 5-10 years, sort of where's this going?
We already introduced the idea that Lizzie and I are using the power of mouse genetics to try and identify the genes that you need in 3 copies to cause the different aspects of the syndrome, the learning difficulties, the early onset Alzheimer's disease, the congenital heart defects, and so on. And so my hope would be in the kind of medium term, meaning 5 years, I don't know, that we will have identified quite a number of these causative genes. We will know that it's gene X present in 3 copies. You need that in 3 copies in order to have learning difficulties. Gene Y for the heart, gene Z for the early onset Alzheimer's. So identifying the genes is sort of step 1 for us, if you like, although it's a very long, it takes a long time to do, but that we think is a very important aspect of it. And then to combine that with analysis of what's actually going on at all levels, the whole animal, the organ, and the cell in particular, to understand what are the changes that are, how is it that 1 extra copy of gene X is causing congenital heart defects or whatever it is it causes. So understanding the pathways, the biochemical, essentially pathways that are changed as a result of that. So those would be my hopes for the future. Those would be the discoveries that we might make. Why is that interesting or important? Well, I think it's interesting per se. I mean, I work in this because I think it's an interesting problem to work on. But I think it has immediate potential application, which is that if you understand how something goes wrong, you can think about rational ways to make it not go wrong, to fix it, if you like.
Are there people within the sort of consortium who would take on that bit of the challenge, or would that be kind of bringing in?
I think it depends on what it is we find. The likelihood is, if what you need to do is to develop a medication to treat, to interfere with a particular pathway or to fix a particular pathway, that's going to be large drug companies. We're not drug companies. We can't screen millions of compounds and find things. But what we can identify is potential targets, which can then be taken on by others. That would be the way that I would see it. We wouldn't invent the medication. We would set the stage where others can then follow and use that information to design potential therapeutic approaches.
And so
there's a question here.
Do you think in the future that they'll be able to do in utero modification and get rid of this third gene rather than...
Get rid of the extra gene?
Yes.
Or genes?
just the chromosome?
The chromosome.
Yeah, well, all of the chromosome. I think that's, in principle you can imagine such things. I don't think we don't have the technology to do it. So it's not feasible at the moment. I mean, I don't know how many cells we have in the body, it's 1 trillion or something, and to change the genetic makeup of every cell in the body is completely beyond us at the moment. We can't even imagine how to do it. Whether it's possible to ever do therapy in utero either, I don't know that either. This is not an easy place to do therapy in utero. But it's not clear that you necessarily need to do it that early. It could be after birth, for example, the treatment, or even, well into someone's life. And so there have been recently, a set of studies that were done in one of the mouse models of Down syndrome with a learning and memory test, interfering with a particular pathway in the brain. And the effect of this was being done with adult mice who already had the learning difficulties. And you could reverse their their memory deficit by giving them this particular drug. This is a drug that was already FDA approved. It didn't have to be developed. And the effect persisted, in fact, for a couple of months beyond taking them off the drug. This drug is now, or similar types of medication, are in clinical trials at the moment. I think Roche is currently trialing it. So we don't know the answer to whether it actually works in humans yet or not. But actually that was quite a bit of a turning point, because I think people had seen Down syndrome as a condition that is set during early embryonic development. It's something that happens in utero. By the time the baby is born, it's too late. This suggests, the mouse is not the same as the human, but in the mouse you could reverse this, or at least reverse one aspect of the syndrome, this particular memory problem. So I don't think it's impossible to imagine that you could do the therapy in adults or children, after birth. But to remove the genes, I think, the extra genes in utero, I think at the moment we don't know how to do that.
Well, so Andre, I've kind of got two final questions for you. One is also the question of what advances you'd like to see coming up in the future, but also what is the current research? Is that impacting on the people you see in the clinic? Is simply the understanding of what's going on, is that having an impact now?
I mean, I do think that the work that we do will, and already is having an impact on the work that we do in the clinic. So, for example, by understanding what are the earliest changes associated with Alzheimer's disease in people with Down syndrome through these kind of tests and things that we do, will help us, or is already helping us, to develop new diagnostic tests that we can use in our clinics. and that will help us to push the diagnosis earlier so we can identify people at a much earlier stage who are beginning to show the early changes. And that's very important because we already have some symptomatic treatments. So these are treatments that don't necessarily change the course of disease, but it can improve some of the symptoms. And it works very well in people with Down syndrome. It does help to slow down some of the development of symptoms. But we're hoping, through some of the work that Lizzie is doing and the geneticist John Hardy and the cellular scientist Dan Nishtik is doing, that we'll be able to identify potential new treatments. And the treatments can be of two types. It can be treatments that help to prevent difficulties before they develop. Or it could be treatments that we can give once people have started to develop problems to stop the progression. So the hope is that we would eventually have treatments that we could use in both those kind of ways. And in many ways, people with Down syndrome stand to benefit most from those kind of treatments because of the high risk. And they also probably one of the best populations in which to eventually trial safe treatments such as preventative treatments.
I'm going to come out to the questions from the audience. Before I do, I'd like you to... We'll do a thank you at the end, but I think a thank you at the moment as well, so it's not speaking. So, who would like a question? So, we got shooting up everywhere. So, can I come to you at the front?
Just a technical question on the mouse model. How is it you engineer the full or partial tracer in the mouse?
How do we do it?
How do you introduce the
Okay. Well, the key to it is a rather special type of cell called an embryonic stem cell, for which the Nobel Prize was awarded in 2007, I think. So developed by Martin Evans in Cambridge. This is a type of cell that you can grow it in the Petri dish forever, essentially, but it has a remarkable property that when you take these cells and you inject them back into an early mouse embryo, this is a pre-implantation embryo when it's still a small ball of cells with a little hole in the middle. You inject the cells back into that, put that embryo back into the uterus of a foster mother. The mouse then carries the embryo to term, the pup is born, and the cells that you've injected end up contributing to all the tissues of the mouse that is born. Now, this is a route of essentially turning a cell in a dish into a whole mouse. And then any genetic change we can make to the cells ultimately is transferred into that mouse that is born. That's our route through to the mouse. And then what we do, so it's taking a step back, how do we do it? So we have to do the genetic change not in the mouse, we only have to do it in these cells that are grown in Petri dishes. And that we do by a variety of methods, but basically we're introducing pieces of DNA into the cell by an electric shock that the DNA goes in. We make that piece of DNA recombine into the correct position in the genome. And to make the duplications, we do that twice at either end of the region that we want to duplicate. And we've inserted at either end special sequences, DNA sequences that can be recognized by an enzyme that recombines them. And if it's done across two chromosomes, the recombination ends up either creating a deletion or a duplication. In fact, we usually get both events, so we can make mice with both events. So we use a combination of kind of enzymes and DNA in these embryonic stem cells, and then we inject those cells into mouse embryos, and from that we get the mouse strain.
Are the animals able to reproduce, or do you have to
Yes.
continue to say, okay, cool.
So all the strains that we've made are viable. Some of where we've tried to make large deletions, they tend to be rather infertile. So having extra genes is usually not too much of a problem. I mean, if you saw the mice that have the extra duplications, you wouldn't be able to distinguish them in the cage. They look identical. You have to do, careful tests. Their memory may not be quite as good, but in a cage they look essentially identical.
And finally, of the 300 approximate genes that you look at, are they all protein coding genes or more?
Well, I
microRNA,
think I used the phrase depending on what you call a gene. So that was 300 protein coding genes. So there's other stuff there. So our view of what is a gene has changed. We used to think that most genes had information to code for a protein, to make a protein. We now know there are lots of so-called non-coding genes. There are actually probably lots of other things there, and they're not terribly well characterized. So we're hoping for our mapping project to identify the gene. We're hoping it's not one of those rather obscure, ill-defined genes, otherwise we'll be looking for quite a long time.
So you've still got all the data in case anything else new comes up, and you can maybe refer back to it, the data set.
Sorry?
Have you taken a wider data approach? Then, if anything new comes up that might be implicated, you can look back in your previous experiments.
Sure. Yeah.
Pass the microphone directly behind you.
You have an extra gene, chromosome, a third part. When there's a question of transposition, that would be the opposite of the problem. There's something missing, and that's why children are being aborted with gross problems. Is that anything that parallel world where this is being researched, and could that be helpful to you in your research?
There are many different human syndromes where people have difficulties of very different sorts because they have as what we would call monosomies. So we call having the normal pair of chromosomes would be a disomy. Down syndrome arises from a trisomy, having 3 copies, but if you have 1 copy of a chromosome, not a gene, as you rightly say, a chromosome with multiple genes, that's a monosomy, and there are many conditions like that of partial monosomy. And the sort of technology that we use could be used to investigate such syndromes. That's exactly right. So what we do, we call chromosome engineering, and you can engineer chromosomes to have extra copies or reduced numbers of copies to go from not the normal 2, but up to 3 or down to 1. So it is being used to investigate those disorders, but those disorders are much rarer than Down syndrome, and so there is simply less research going on
less
those.
research,
Yeah.
Okay.
But I think it's an interesting question you bring up about you said abortion. I guess you meant miscarriages.
Yes,
maybe.
I meant that.
Yeah.
Spontaneous abortion,
And
yes.
it's a statistic, I think, don't know how well founded it is, that about 70% of Down's babies actually miscarry.
Oh, really?
So what is born is, if you like, not even the majority of them. And where you get, and so people often ask me, well, why trisomy 21? What about trisomy of all the other chromosomes? Why don't you see that? And the answer is they almost certainly are made, but they all miscarry spontaneously, with the exception of, I think, 13 and 18.
13 and 18 rarely will go through to term, and children are born with trisomy 13 and trisomy 18, but they usually die. I think trisomy 18 is usually up to the age of 5, and the children die before the age of 5, and trisomy 18, the children die within the first 6 months to a year.
Oh yeah, sort of related to the last question, I guess, but is the mechanism by which the third chromosome happens to be there, is that understood, and is it similar in humans and mice? And does the process of how you induce it in the mice give you any clue as to how to fix the problem in humans?
How we do it in the mice is we do it deliberately through this sort of genetic engineering technology in these embryonic stem cells. So we've engineered the mice, and every one is born with that extra piece of DNA. In humans, it's the reasons for getting the extra chromosome are completely different, and they're to do with an error during the making of either an egg or a sperm, but usually an egg, as it happens. And so humans have 46 chromosomes, there are 23 pairs. During the making of an egg or a sperm, you have to divide that down into just 23, one of every chromosome. That's a process called meiosis. And that division process occasionally goes wrong. So instead of the cell dividing and sending 23 chromosomes one way and 23 the other way into the 2 daughter cells, occasionally 2 copies of the same chromosome go into the same egg. So if an egg is carrying 2 copies of chromosome 21 and a sperm comes along and fertilizes it with its own 1 copy, the embryo that is the fertilized egg then has 3 copies of 21. The reverse could be true. It could be a sperm that has 2 21s, but we know from various genetics that it's usually from the egg. And in fact, you may well be aware that by far the highest risk factor for Down syndrome is maternal age, is the age of the mother. And so it seems to be something, not entirely understood, correct me if I'm wrong, something about the age of the mother and actually therefore the age of the eggs that are made that predisposes to these chromosomal errors, these errors in segregations when the cells are dividing. So when a woman is born, she already has, I think, all the eggs that she's ever going to have are already there, but they're poised halfway through this meiosis process, halfway through the process of doing this segregation, of separating the chromosomes. And then during ovulation, that egg matures and does the final division, and the chromosomes segregate, and then the egg is ready to be fertilized. And I think it's that point that an error creeps in, usually.
Obviously, in terms of the research, what you're doing on Downs is all-encompassing and covering the whole spectrum of Downs. But if you look at it from the other perspective, from Alzheimer's, how significant is Downs research to Alzheimer's?
Well, I'm just trying to think. There's two ways of answering this question, isn't it? First of all, Lizzie mentioned John Hardy, who discovered the link between the APP gene and Alzheimer's disease, and he partially discovered that by looking at the genes and the cells from people with Down syndrome. So that helped to make that link. It is complicated doing it the other way, but we do learn what is happening in the general population in Alzheimer's disease research. And at the moment, for example, we're very interested in comparing families with very particular risk for Alzheimer's disease due to duplication or triplication, if you want, of the APP gene. So there's a small number of families with very high risk for Alzheimer's disease who have that particular problem. And that, we believe, should be similar to the Alzheimer's disease that we see in Down syndrome. But there are some important differences, and those are key differences, really, to understand what happens in the rest of chromosome 21, what helps with the protection, for example. So we can do things like that. A lot of the research is focused on familial Alzheimer's disease, so people who carry very specific genetic problems that increase their risk for Alzheimer's disease. It's a bit more difficult looking at sporadic Alzheimer's disease, which seems to occur without a very strong family risk, although there is some family risk. But we can use some of the techniques that they're using as well. So we can use some of the neuroimaging techniques that they're developing to mark the plaques that develop, the amyloid plaques. So we can use those kind of techniques in people with Down syndrome as well in the long run, we hope.
I'm just going to jump in with one further comment about that, which is one of the experiments that Victor and I have done is we work with a mouse model of Alzheimer's disease. It's got nothing to do with Down syndrome. It was produced many years ago by a group in San Francisco investigating Alzheimer's. They're not interested in Down syndrome. So we have this particular mouse model. And what this mouse does is it lays down the protein in the brain that you find, one of the proteins in the brain that you find specifically in human Alzheimer's disease. So we've been able to take one of our mouse models of Down syndrome and cross this, so to mate the two types of mouse, the Down syndrome and the Alzheimer's type of mouse. And we've got, therefore, an animal model which has the propensity to get Alzheimer's disease on the Down's background, if you like. That's one way of thinking about it. And that gives us a severer form of Alzheimer's disease. And so that's of great interest to the Alzheimer's community because our starting point there, in one way, was an Alzheimer's mouse being modulated by genes on chromosome 21. And so, again, from the Alzheimer's perspective, if we knew what those genes were, that would be very important for understanding Alzheimer's disease. Because even after we've discovered, or John Hardy found, the first genetic mutation ever that's known to cause early onset familial Alzheimer's disease. Just straight, if you've got that mutation, you get Alzheimer's disease. We don't actually know why. And that was in 1991, so it just shows how difficult it is. Even when you found the gene, you know the genetic mutation, we could read out the base pairs, the DNA sequence to you. We could not tell you why that mutation actually gives you Alzheimer's disease. It's very difficult to then do the cell biology, so we hope these mice will help.
I would Can I give a third different answer? Because you've actually touched on quite a lot of interesting issues in that question. So I think another interest from the Alzheimer's community or from the pharmaceutical community in the Downs population is that there have been quite a number of clinical trials trying to cure Alzheimer's or treat Alzheimer's. And by and large, correct me if I'm wrong, they've been abject failures. So despite a lot of research and a lot of effort, there are no effective treatments that make a big difference, certainly none that reverse the condition. And one possibility for why that is not that the therapies and medications that have been generated are barking up the wrong tree, but that they're treating people too late. They're treating people who already have dementia, often quite severe dementia. And we know that the natural history of that dementia is that they develop these depositions of these amyloid proteins years and years earlier, possibly 10 or even 20 years earlier. And it may be that you have to treat them then. And by the time the symptoms arise, it's way too late. All the damage is done, essentially, and you can't reverse it. That's one possible explanation for all the failures. In which case, you'd want to treat earlier. But how are you going to do that in the general population when, I don't know, the fraction of people that end up with dementia is, 5%, 15%, I don't know, by age of 80 or something. Are you going to take a lot of, you're going to take 100,000 individuals aged 50, treat them all in the hope that, amongst those there would be people who would have developed Alzheimer's. You have to do very large studies. That's very expensive, difficult, long-term. You can't predict ahead of time who's going to get the dementia if you have to treat ahead of time. The Downs population potentially offers a different route. This is a population within which we know 50% will develop the symptoms at a particular point in their life. So you can treat them 10 years earlier, 20 years earlier even, if that's what's required. And then, it's a long clinical trial, you've got to wait 20 years, or 10 or 20 years for the answer. But at least then you know that 50% will get it and 50% won't. So there's a lot of interest in whether the Downs population could be used for that purpose. Of course, they would be the first beneficiaries of the treatment as well, but it might be useful for everyone else too.
I just wondered what research with regard to the accelerated ageing that you see in some people with Down syndrome, and whether there's any chance of learning from that for that condition where people age very quickly, progenia.
There are groups, I think, that are interested in some aspects of accelerated aging in people with Down syndrome. I think the cellular researchers are particularly interested in that because they can see the similar sort of aging effects at the cellular level. And so there are some cellular research groups that are already looking into that. And it's possible that that sort of accelerated aging effect could contribute to the risk for Alzheimer's disease in people with Down syndrome.
So we're going to have to wrap it up there, I'm afraid. I would like to thank you all for coming. One final thank you to our guests, and please have a drink after this statement. They're here for a little while afterwards if you want to ask them a question that you didn't want to ask in front of the whole room. So thanks very much.